of uh, why we can put keys someplace, or I'll put my glasses down and I can't remember where they are. But if I put them in the right place, put them in the same place every day, I can have long-term memory and check it out. So olfactory organs, very, very important, particularly if you're in a house with a gas leak. I mentioned that before, but that's my favorite example. So natural gas has odorants, smelly things put in. So if the gas is leaking, you can sense it. Also, I wasn't aware of this. When you burn natural gas on your stove or your hot water heater, a lot of pollutants and dangerous chemicals are produced. But that's what we've been doing to the earth, polluting it and polluting the earth and polluting ourselves. So vote. Please take care of climate change. I want my new granddaughter to be able to grow up in a clean world. Here's the structures. These are called conchi. They are allow for air to enter through your nares and circulate around these ridges and valleys and allow your air to be warmed. Your olfactory nerve is just above your nasal cavity. It sends nerves into the just above the nasal cavity, and materials can be recognized, and that information is sent to the olfactory center through the olfactory tract, and it's very important. If we look at the fine structure of the receptors, we have supporting cells, and right here, and then we have receptor cells that have, and these are bipolar cells. You can see that they respond to odorants, and somehow the odorants cause action potentials in the cells, and that's how we can be aware of what's around. So let's look at the pathway. Well, we didn't see it, so anyway, we'll have the starting in the nose. The nerves send information to the olfactory bulb, and olfactory projects to the cerebral cortex, the olfactory, the hypothalamus, and the limbic system. The hypothalamus and limbic system are parts of the brain that we share with all animals and are very, very old systems evolutionarily. But again, smell is very important. Taste. We have taste buds all over our tongue. We can sense salt, sweet, sour, and bitter. And there's a new sense called umami, which is a flavor apparently in Oriental Japanese cooking. And the taste is going to synapse in the medulla oblongata and send information to the gustatory center. So gustatory receptors are mapped on the tongue. Certain tongues sense umami, sourness, sweet, and so on. This is a microscopic image of the taste buds in your tongue. They respond to chemicals in food and send a message and detect umami, sour, bitter, salty, sweet. So the gustatory receptors have microvilli, taste hairs, and when chemicals dissolve around these hairs, nerve messages are started, and that sends information, and we're able to recognize various chemicals. And we all know that taste and olfaction go together. And why the heck do women smell better than men, or because they wear perfume, or why do they have my wife is all kind of time smelling something that I have no idea what she's talking about. Okay, so olfactory information goes directly to the cerebral hemispheres, can affect mood and behavior. Uh, Gustatory sensations are integrated with smell. The eye. So let's look at a structure. So we have the eyelashes, the eyelid, the palpebra. We just got to know, what do we got to know? Sclera, iris, pupil. That's it. So somebody coming in. Yes, we are. Welcome. Okay, we're back to the PowerPoint. So let's keep going here. You should know what a lacrimal gland is. Lacrimal ducts, which dump into the lacrimal sac and drainage into the nasal cavity. So it's very important that the lacrimal gland produces tears to help keep the surface of our cornea lubricated and protected, because any dust that gets in your eyes or any bacteria hopefully will be washed into the lacrimal canal, into the nasolacrimal duct, and into your nasal cavity, and you'll get the sniffles or you'll have nasal drip. The intrinsic eye muscles are 6. We have the lateral rectus, the inferior rectus, and the superior rectus. So that will be 2 lateral rectus. There'll be 1 inferior rectus, and there'll be a superior oblique, and all of those muscles are involved in eye movement. If we look at the eye from the anterior, superior rectus, inferior rectus, medial rectus, lateral rectus, and an inferior oblique. So 5 muscles, not 6. The eye has the eyelid and so on, and we have the fibrous layer, vascular tunic, and neural tunic. There's an anterior cavity and a posterior cavity. There's a gel in here that protects the retina. The vascular tunic includes the ciliary body, the suspensory ligaments, and blood supply in the choroid. The fibrous tunic is very, very tough and helps maintain the shape of the eye. The neural tunic is where the retina, the neural part, and the pigmented part, which helps reduce bright light and prevent scattering of light. So all I want you to know is the anterior chamber, the posterior chamber, ciliary muscles, ciliary body, lacrimal sac, and the muscles. So the fibrous tunic is called the sclera, and the cornea is transparent. I mentioned that this fibrous tunic is very, very stiff. If we had the opportunity to do the eye dissection in lab, but we're missing that, so you just have to take my word for it. So we mentioned the vascular tunic, and that includes the ciliary muscle and ciliary processes attached to the suspensory ligaments, and that helps change the shape of the lens based upon whether you're looking at a near object or a far object. We talked about this, vascular tunic, and then what does the vascular tunic do? Carries blood vessels, adjusts the diameter of the pupil, and also the shape of the lens. The pupillary muscles, pupillary muscles, and they either enlarge the size of the pupil or decrease it. So in very, very bright light, everybody's seen this and is aware of this. If light is very intense, in order to see, we have to reduce the amount of light striking the retina. If the light is very, very dim, we have to enlarge the pupil so that we are able to get enough light to see. And if it's totally dark, we may not see anything. The neural tunic is the retina. And let's keep looking at the parts. So there'll be a question just like this in WebAssign. So the light comes through first the ganglionic layer, the ganglion cells, the bipolar, and then the rods and cones, and then the pigmented layer, and then the choroid layer. So there'll be a picture like this in WebAssign, and you'll have to identify these various parts. The back of the eye, where the optic nerve enters the eye in order to connect nerves to all parts of the retina, that's the optic disc. There's no receptors with a nerve. That's called the blind spot. We have the pigmented layer. We have blood supply, the optic nerve, the sclera, and then the choroid. So you can do experiments where you identify the location of the optic disc, but we won't get a chance to do that. If you look at a view of the back of the eye, you can see blood vessels. You can see the fovea and the macula lutea. And the fovea is where you have the most cones. The optic disc is the blind spot. And obviously, your eye needs blood supply for oxygen, remove carbon dioxide, provide nutrition for cells. We mentioned these already, go through this. The aqueous humor is the front cavity in front of the lens, flows into the anterior cavity. So just read through this. Look at these structures. Again, you should know the lens, the suspensory ligaments, the ciliary processes, the ciliary muscles, and so on. And the anterior chamber, the posterior cavity, there's vitreous humor in here. There's another humor in the aqueous humor in the cavity between the lens and the pupil and the cornea. Okay, read through that. Let's keep going. So we said that light has to pass through the receptors. So light's going to pass through, and you'll have the ganglion cells, the bipolar cells, and then the light will change the shape of rhodopsin molecules. That change in the shape of this molecule leads to hyperpolarization, and the many rods and cones that we have in our retina allow us to send varying patterns of action potentials to the visual cortex, and amazing we can see. So I can look at this, I can tell this is orange sort of red, grayish, brownish. I have color vision, and I can use my stimulus of the rods to pick up low-level light and help me distinguish between the low light and bright light. The bright light allows me to pick up color and detail. When light hits the rhodopsin and the change in shape leads to, release of energy, and rhodopsin goes back to its first shape, but that change in the shape of the rhodopsin leads to the generation of nerve impulses in the retina. So how does this information get back to the brain? So light optic nerve, optic chiasm, where the nerves cross, some of the nerves cross, and some go to the same side, the right cerebral hemisphere, others go to the left. And then we have the optic tract, the relay point of the thalamic nuclei, the superior colliculus, and some input stays on the right side and others go into the left side to have a depth in our vision. Apparently, this splitting of nerve impulses, some to the same side of the visual cortex, others on the other side, leads to our ability to have depth perception. Okay, the ear. Let's turn off the recording. I went a long time there. So let's see if we can remember how to do that. Stop recording.